أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم إذا الشمس كورت وإذا النجوم انقدرت وإذا الجبال سيرت وإذا الإشار طلت صدق الله العلي العظيم. Dear students, I came with the part 2 of lecture number 2 for second year class, and I will start from sequencing of the DNA of single chromosome. It's very difficult, but even you will know the RFLP sites, still you will get very important information, because genome of human being is 25 times larger than the other. I test you, I check one thing. For example, Huntington disease, you can understand when you know the restriction site position. So even you are unable to calculate the complete sequence of DNA, that is laborious work. Even you know restriction sites, you can know the diseases, because nature favored us. They kept some diseases very closer to restriction site. For example, these are the two DNA, and that is the restriction sites position of this one. And in this one, if the restriction site position in this status, if the restriction site is present, so disease is gene is very closer, doctor will identify Huntington disease will be present. But in this case, in B case, because here there is no restriction site is there, so he will say this man will be free from Huntington disease. So this is a very good learning, that transgenic bacteria are those bacteria because you change the DNA. As I already told you that you can change the bacteria character by using plasmid. You will enter the genes by entering genes by making them transgenic, mean that you will enter the genes artificially from the outside. They will become the transgenic DNA. So transgenic DNA and transgenic bacteria, they are transgenic bacteria especially we can use for many purpose. For example, I will tell you some examples about Drosophila were being genetically engineered. Mouse is genetically engineered. Humans, gene therapy, which I am just going to tell you after some time, because gene therapy is the process by which you can change the genes in the humans. Abnormal genes can be replaced. Transgenic plants, you can, all plant cells are totipotent. Totipotent means they are not much differentiated. So when you are going to enter the genes into the plant, you have to take care. Reason, because you have to expose the protoplast, and by particle gun is being used to change, and there are some other processes by which foreign gene can enter. And especially we are using the vector. Bacteria is a very good source. You can enter the required gene by entering into the plasmid, and bacteria will cause infection or virus will cause infection into that plant. You can make a lot of good crops with good yield, and you can also finish the diseases also. And I will tell you C4 cycle. If you studied in first year, you know in which carbon dioxide will be more available to the ribulose carboxygenase. You understand the ribulose oxygenase enzyme studied in the first year. Ribulose carboxygenase, ribulose biphosphate carboxygenase react with carbon dioxide in the Melvin-Calvin cycle, but it will react more with carbon dioxide. Normal cycle is C3 cycle, you know, but in C4 cycle the more carbon dioxide will be available, and that is also made by you studied C4 cycle, but I did not tell in that time that plant was genetically engineered, and that was synthetic made by scientists. So we can increase the efficiency of the photosynthesis by using the C4 cycle, by making increase of reaction of polyhydroxybutyrate, another type of polymer, biodegradable polymer we can form. That is very simple, because when you are genetically engineered gene which can form a polymer which is a biodegradable, you can use plants, you can use mouse, that will be used, and that polymer will be biodegradable. That is the problem currently going on in the pollution case. So you can use a genetically engineered plants and animals to make the biodegradable polymer as well. No, luciferase enzyme was sprayed in the tobacco plant, and luciferin gene, definitely gene is calculating the production of luciferin, and it will glow by action of a chemical, and that will also is evidence that you can change the characters of the individuals. So if we talk about Arabidopsis plant, but before that I will tell you gene therapy. Gene therapy is the process by which you can change the abnormal gene with the normal gene. And there are two types of gene therapy. One is a in vivo therapy and ex vivo therapy. In vivo therapy, ex vivo therapy. Look at the figure, I will tell you. You are removing the tissues. And removal of the tissues from here, from the body. You are removing tissues, and you are entering reverse transcription enzyme will help. You are entering the required gene in outside the body, that is ex vivo therapy. Defective gene, this is the defective gene, and defective gene was replaced by normal gene by using virus or the bacteria. You use retrovirus cause infection in the human body, so human tissues are enough for retrovirus, it will cause infection, and that normal gene by retrovirus will be entered into the human tissue, and you will transplant once again. This is called ex vivo therapy. Ex vivo therapy is used in different curing of the disease. I will specifically name one disease, SCID, Immuno Deficiency Syndrome disease, especially in the children, and it will not mature B and T cell, which is very significant disease. Bone marrow stem cells, they will form these tissues, so you will use a normal gene which will make this enzyme adenosine deaminase in the bone stem cells, which will form these two cells, the T lymphocyte, the B lymphocyte. They will be matured. It is almost the blood cancer. So this enzyme level will increase by using the normal gene. So that is significant solution of this dreadful disease, blood cancer. I will tell you one more thing. Cystic fibrosis, another disease which is treated by the that is in vivo therapy. In vivo therapy, you will use the liposomic particles in the solutions which consist of normal genes. What is the cystic fibrosis? I will tell you. These are alveoli cells. Alveoli cells has the channel protein which allow the chloride ions to go into the cell. So when chloride ions will not enter, definitely it will cause the disease cystic fibrosis, and that is due to lack of an enzyme which is controlled by significant gene. So normal gene which is controlling this protein channel to for the movement of the chloride ion, you can add very easily by using liposomic particles in a solution and coated by the normal gene. So when you will as an inhaler, this gene or chloride ions, controlling chloride ion, will enter directly into the body, and that is just a treatment of cystic fibrosis. You are directly entering the gene which is controlling the chloride ion. Cystic fibrosis disease can be cured by in vivo therapy. Tissue culture technology mean that you can make all plant cells are totipotent. Totipotent word reason that all plant cells almost have same genetic makeup, because they are not much differentiated as the animals. Therefore, they are the best source to form the seed. Even you will use a tiny piece of phloem, tiny piece of specialized cell, and you can use to form the callus, and many seeds or many tissues can be formed, and those tissues can be packed in the packets and you can export. No need to grow a successful plant seed of the successful plant growing on the land. You just collect small tissues in the laboratory, and you use to you provide amino acid, provide the nutrition, and provide the exactly the same materials which is required by the plants, and separate them. And not only to form the seeds, you can make cell suspension. For example, if any plant is very useful to get the food, to get the disease medicine, so you can use tissue culture technology to produce the suspension in the laboratory, because in the laboratory artificial seeds, as I already told you, you can make the artificial seeds, you can export them, you can ship them. They are not being grown on the field, they are normally growing in the laboratory by using small tissue in the laboratory. Tissue culture mean you will get small cells or small group of the cells from the plant, you will take them into laboratory, provide them the nutrition and grow them and use according to your need to form the medicines in the form of suspension, to form the seeds, to form the seedlings, small plants, and save them and export them to form the vitamins. Anther culture is used for different type of the flower, because different flowers have different benefits. If one flower is monopolized, that is more beneficial, so you can use that anther culture by using by changing the number of chromosome in the sperm, and now the zygote will be formed according to your need, and that flower will be total beneficial for you. Similarly, genetic engineering, I am talking about genetic engineering of the plants. Those are almost same. DNA can enter by highly electric pulses. You will enter, because DNA enter through small pores in the cell membrane. And DNA luciferase enzyme, that was the point I was talking some before. Enzyme luciferase was inserted into tobacco protoplast, and that was grown by the chemical luciferin. How it is this enzyme is entered by using a vector. You know, the infection is called tobacco mosaic virus. That virus will carry this enzyme in the form of the gene that is inoculated. Or you can change the protoplast at the cellular level during the growth at zygote level, then you will grow. That is transgenic plant you can make. Transgenic animals are very useful, because transgenic animals they are forming the medicines in their milk, they are forming the medicines in the urine, which is not mostly likely procedures among the scientists, but I will tell you how these transgenic animal will help. Human gene, which is very useful gene, and you want to form something, you will enter that gene into the egg taken from the goat. Clear? And egg is technically you used egg. Okay, now this egg is, you entered the gene. Now this egg belong to goat, gene belong to human body. You got the idea? You entered this egg into the goat. Clear? Now this, in the uterus of this goat, the egg is behaving like the zygote with a diploid nature. It will grow mitotically. It will give birth to a transgenic goat. Transgenic goat having genes of human body, because egg was only using cytoplasm. The egg was used. Technically, nucleus of the human gene was used. You got idea? I will explain once again. When you will use, I need to start from here, because that is very important. Where is the indication? I just want to give indication. Indication. This DNA is diploid. I used egg only. Egg belong to goat, and DNA belong to human gene, which is diploid. So diploid nucleus in the egg behave like zygote, because zygote is diploid. You will enter into goat. The goat will develop. All characters in transgenic goat will be from the human, because human gene, if the producing insulin, this goat will produce insulin, will come out in the form of milk, in the milk or in the secretions. You can calculate. You can increase this number of transgenic goat to n. You take the cells from this transgenic goat further, and you remove the nucleus and entered into the egg once again by using the egg of the another goat and enter these enter these eggs into another goat, and what will happen, the development will take place, these eggs will develop in these goats, they will give birth to the transgenic goats, and goats number will increase. I will explain once again from here. This is the transgenic goat having human gene. You just get one diploid cell from that goat, you remove the nucleus, you take another goat which is not transgenic, you use egg, you inject the nucleus of this transgenic goat into the egg of this non-transgenic goat. Now these eggs behave like zygote, they will enter into the womb or uterus of the normal goat, and this goat will give the birth to the transgenic goats, and this transgenic goat will further increase the secretions of which you want. So how these transgenic membranes and transgenic animals and the plants, they are helpful. Even I will tell you Arabidopsis plant. Another way is that Arabidopsis plant, some plants problem is that they will not grow in the salty conditions, in the salts conditions. So when they will not grow in the salty condition, so you can use the vacuole, you can use the vacuole, and when you can use the vacuole membrane, there is a gene you find out, it will increase the permeability of sodium ions into the vacuole. To just make Arabidopsis was made transgenic by adding gene which will increase the sodium ion permeability to the vacuole. So when this plant, transgenic plant, Arabidopsis or any other plant, experiment made upon Arabidopsis, when it is used, it will be grown in the salt tolerant crops, which is almost many percentage area in the Pakistan which is not being grown, and that is just like a barren area which is not being grown and not fertile area, and soil, lot of the soil field are being wasted by this way. So this by making transgenic plant by this way, sodium salts which are more harmful in salt tolerant crops, they will enter into the vacuole, and this crop will become salt tolerant, yani this plant can be grown into the salty area as well. So this is the output of the biotechnology. Second year class, this was the use of the biotechnology genetic engineering, part 2 of lecture number 2 I explained. Thank you very much.